to the bench link. All right, so just upload them upload all the plasmid for now. And let me see. Let me try upload it too. I don't have it on this computer. Okay, so once you get that, you could go into your benchling count. All right, so you should go into your benchling. It depends on what you have. I already have some sequences up. But you can start importing them. So you want to add a DNA RNA sequence, you can import, right? So you could choose your file. I'm gonna import a PS, right? Okay. All right, so that's upload is done. I should have a PS. Okay. So here would be the PS, right? You could go to the linear map where it could give you all of the where each site is, or if you want the plasmid map, here it is, right? It could give you a description. Hi, copy episomal. That's important. Forgot to mention it. Episomal, remember I said episomal is when they can go into the chromosome. So it's a galactose inducible expression of protein in, these are for yeast, so Saccharomyces cerevisiae. This is my favorite vector. All right, it's my child. So it has an episome actually for the uracil. So let me see, gal promoter. They don't mark it here, but there are, where is the border? I don't think they mark it here. No, they never mark it, but there should be a left and a right border. Or is it from the uracil gene? There should be a left and right border that allows the episome, allows it to be an episome. But anyways, we don't need to talk about that one. All right, then you insert the other one. Import the next one. I'm going to import PUC. PUC, I think it's for, PUC is for bacteria. Oh, it could be for phage too. Yeah, it's an E. coli vector, multiple cloning site for DNA cloning. But it can be used for phage as well, right? That's M13. M13. Even have the person mimic it. All right, then I insert the next one. So just insert all of them right now into your benchling. Sorry. I'm gonna insert the last one, pick Z. Pick Z is also for yeast. This one is Jan's vector. No, Jan used, what did he use? I forget what he used. This is for Pichia pastoris. It's also a yeast. It's, yes, this one he used this. He is Pig Z. Pig Z, I am not sure it's beta or C or alpha he used. But you would have to feed them methanol. For PS, you feed the yeast galactose. For Pichia pastoris, in this specific plasmid, you have a AOX1 promoter, which is turned on with the presence of methanol, right? So once you feed it methanol, it's gonna start secreting your proteins. Okay, so now I have all of them here. Let me bring Picambia up here, right? Okay, now I have 4 of these vectors. Let me do the plasmid map. just so I could look at them better. Okay. Now you also are given a sequence, so the DNA to be used, right? Okay, stay with me, stay with me. All right. What do we import from the drive? You import from the drive. Sorry, guys. You could turn on your mic because I'm not in the virtual, right? But you would import pCambia, PIXZ, PUC, and PES. All four vectors. Import them into Benchling.
Miss, the
Yes.
file you shared with us only has kinetic master. It's only the kinetic master folder, not the entire
Oh, I didn't share it. Yeah, it's on Moodle. You want I share it on the chat?
Yes, miss.
Okay, hold on. Copy link. Mm-hmm. Mm-hmm. There. Go into that folder now. I will wait for you guys. I'll give you 2 minutes. Time is of essence. Ay. Hmm. I'll need to give you your test soon. I can check. But don't take away our class from me Thursday, but we are meeting at the night. I am not going to miss a class on Thursday, and then Friday we will still have lab because we can't afford to miss a lab. Because me a Hicks. Um, let's see, let's see. Everybody got it? Edwin said he really lost. What happened, Edwin? Let us help you. So, like I shared this here then. So go to this drive, right? And what I want you guys to do to download the file named pcambia, ppixza, puc19, and ps2. Download those files. Yes? Don't worry, we have 2 hours to get this. I need a snack. I should make all of you guys turn on your cameras so I don't feel like I'm talking to myself.
Miss, just run all of them, right?
Sorry?
Just open all of them on the bench link, right?
Yes, import them. So like how, let me show you guys, right? So for to import them, you would click the plus button on the left hand side called create, right? So you press the plus button, you go to DNA/RNA sequence, you go to import DNA or RNA sequence, and then when you click that, I just share my entire screen. Hold on. Okay, so I think now you're seeing it. So you're going to create, right? Press the plus button here. Go down to the DNA RNA sequence, okay? And you're going to press import DNA and RNA, and this should pop up. A new window should pop up called upload files. You're going to choose a file, right? For example, you could choose PS, right? So you're just going to double click and choose that. It's going to load, okay? And it's going to tell you, oh, it finished load. Upload is done. You just press the close button, right? And then it will, bam, see there? Yeah, appear, okay? That's all you have to do for the vectors, right? For all the plasmids, sorry. Let's see what we could. They will have CRISPR, but we'll not get into that. I'm not really good with CRISPR. Everybody got it? All right, cool. If you don't get it, just let me know. Which file do we upload? You will upload this one. Upload pCambia, pPixZ, PUC, PS. All the vectors that's there. 4 of them. Upload them, right? One by one. pCambia, pixZ, PUC, PS. PS. Don't ask me why they name it like that. They just feel like, I guess. I'm not sure. And I think it more has to do with where they isolated it from. I have no idea. But yes, upload, upload. Let us know if you're still wondering what's going on, because we have to be on the same page here, literally. Let me know when you guys are done.
Maybe we can comment down in the chat to see who I'll finish, and then the rest maybe you can assist them if they can't do it.
Okay, that's a good idea, Myra. So comment done when you're done. Only 4 people. 5, 6, 7, 8, 9, 10, 11, 12. Oh God, I lost count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 16? I think that's okay. I guess that's everybody. I only see 14 others here. Oh, 23. Let's go, guys. We need more. Okay, count again. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Let's go, let's go. 5 more people. Oh, he's still working on it. 1 done. 2 done. Don't worry if yours. Just let us know if your internet is slow. Like my internet here is a little bit weird.
Miss, can we close the one that was there when you open the program?
Yeah,
Or
you
will
can
we
close
need
that one. You can close all the other one if you want. You could always select it later. It's already in file. You could delete that too. But just close it. We don't need that one right now. Well, ever. Um, so you could delete that one. Okay. When everybody's ready, then we'll go to the next step. See, that's why we show me there in a class. Joseph, stop the coming and out.
These sequences are built on Snap. What do you call it? Snap Viewer.
Snapgene?
Hmm.
Snapgene just um Snapgene, all it does is it put the sequence together, and because you could download these sequences from anywhere, the ATG, the DNA sequence, but
Yeah. Yep.
Snapgene just make it look pretty, you know. Um,
Oh, okay. So that
yes,
it's more like of a compiler in a way.
yes, because you can just copy and paste a sequence, and you can put it onto the same bench link, right? And if it recognizes certain sequence, it will just pop out for you. But for me, it's easier to just import than copy and paste, as I lazy. And plus, if I don't want to open benchling, but I want to know what's my DNA sequence or how it looks, then I just double click and Snapgene it'll come out instead of one long 18,000 base pairs of ATGC. You know, it looks more organized. So it just helps you visualize the plasmid. It's only the files we have to upload. The files. Yes, you just have to upload the vectors, plasmid, sorry. Pcambia, P, PIGZ. I always don't pronounce the P in front of PIGZ, but PIGZ, PUC, and PS. There's other cool vectors out there, very fast. Which one was that one again? I have to remember the name. For E. coli. Shucks. What was that? PCR8. PCR8 is the easiest vector, I swear. You don't even need to do any of these craziness right here. But usually it's good to learn because not all vectors are easy to use. I call it vector. They are plasmid vectors, but I don't want you guys to confuse that with the actual vector, which is the bacteria or the fungi or the virus or wherever you're putting this plasmid in. Okay, all right. I think that's everyone. All right, guys. So the next thing to do is to open up the file called DNA to be used. That's a Word file, Word doc, right? So it should be something like this, right? So this is a sequence with 1,152 base pairs, okay? I've colored ATG at the beginning, so it has a start codon. I already put your start codon for you. I chose the stop codon TGA, right, or UGA in mRNA. So I want you to now copy this sequence and paste it onto the bench link. So you just have to go in here. Don't copy it from OD. Copy from ATG all the way to the last A. Copy. Right. Let me share my entire thing again. Hold on. I also don't like share entire screen because of this mirror effect it has in the beginning. It trigger my eyes. Ugh. Okay. So copy, right? Copy this sequence. You go into bench link. You're gonna create again, right? And you're gonna add DNA sequencing, and you're going to say new DNA. Okay. So it says create new. You can name it. All right. I'm gonna name it OD. Okay. I'm gonna say it's linear. All right. I have no idea what's that, so I'm gonna left that right there. It is DNA, and you're going to paste. So that's all of your sequence. And you're going to press create. All right. So now, see, it has the sequence called OD. All right. So from ATG way until TGA. All right. So do that now. So you will have a tab called OD. All right. Whoosh, I hate this. Right. Let me know if it's confusing.
Miss, this topology is linear, right?
Yep, linear. Topology is linear.
And for the schema, we just don't put anything in there.
Yeah, just don't put anything.
When I did mine, I got two. I don't know if you could call it windows. I guess this one that you're showing, and then another one.
Another
On the,
OD?
no, it's well, yeah, I guess, but no, it's like a more like a graph. I guess I don't know.
A graph? Oh, it shows you another sequence of the, I think,
Yeah.
like I said, I could have it.
Oh, it's the linear map and the so this one I have. I think it's the sequence map, and then I got a linear map. Yeah, I think I got a linear map also.
Okay. Well, you could just close one of them because maybe sometimes when you, it might want to show you the linear map. Um, but just go to the sequence map instead of the linear map because we have to create our primers in a bit. We're done. Here comes the hard part. You have to be prepared. And I'm not going to use OD as an example.
I got the same that Edwin was saying, but if you want to like separate them, you just go to the bottom right corner where it has a little question mark and you put split workspace.
Oh, you guys
Yeah,
have.
that's what I
Oh.
did. You could eat for the other ones too.
Oh, no. No idea where you guys are at, but
It's on the bottom right.
I trust you guys will figure out your way. Oh, you guys have like this. Oh, okay. I can't go to my FAA. Wait. Hmm. So I'm just waiting for everybody. Let me. Oh, I don't have it here. I didn't. Hmm. Hold on. I'm going to my next laptop. Let me know when you guys are good to go. I'm just gonna upload another sequence for myself. Done? Okay, everybody says they're done. All right, guys. We need to be on the same page right now. Okay? Because this is going to be a little bit confusing. So please, everybody, please type done when you're done. Okay, I see some dons happening. I think, okay. All right, okay. I guess I could continue. Okay. All right, guys. So now what I want you to do is to insert this sequence or your OD. I have another sequence open up. OD stands for oleate desaturase. It is an enzyme I worked on before. So I'm giving you guys to work on it now. It's a troublesome enzyme. I hate it. But right, oleate desaturase, this is a sequence. I want you guys to insert this sequence into a specific vector. So you have to choose a vector, all right, that you're going to insert the sequence into. Now, the thing is, in order to insert this into a sequence, so let me go to my little chart. Okay. So in order for this to be inserted into a sequence, right? Each vector map, okay, each of this vector map, they are circular, right? So we need to open up this circular map, basically means to cut it and insert this sequence in, right? So one way to cut open this sequence, right, and create what we call sticky ends. So sticky ends are the restriction enzyme that cut this way. If you have a blunt end makes more mistakes. Let me just tell you guys that. Blunt ends make more mistakes than sticky ends. So oftentimes we like to choose restriction enzyme sites that create sticky ends, okay? So when the enzyme would cut, right, so these restriction enzyme cut, they create these sticky ends. So the DNA will split, right? Forming these what we call overhangs. Now, when they split apart, they're gonna split up. So they're gonna split this way, right? And the other one is gonna be like this, right? So they're gonna split up. Now, our sequence that we want to insert, we want it to, so let's say we have a sequence we have here, right? On the ends of the sequence, we want to add similar DNA, right? So that instead of sticking back to itself, it's going to stick to our DNA sequence. So we need to be able to create, right, a site or create the DNA base pairs that aligns with this restriction enzyme site. Okay, basically we need to insert the same restriction enzyme site at both ends of our sequence. All right, and to do that we would use PCR, right? Primer design and PCR. Okay, so it's quite easy. I think we haven't gone through PCR, but basically what PCR does is it uses primers to copy DNA, right? Every time, let's say this is my DNA sequence and I want to copy So this is a long DNA. It stretches until forever. Well, not forever, but just goes on and on, right? But if I only need this sequence, so I'm just going to draw the double strand because they are double stranded. Right? This is going from 5' to 3', and 5' to 3'. Okay? And of course, it goes on, right? It's a long DNA strand. But I only want this particular sequence. So in order to do that, right, when replication occurs, they usually have set primer. So if you've gone through your DNA replication, I'm not sure if, yes, you guys have to take cytology as chemists too, right? I'm not sure what type of biology courses you guys have taken. But in order for replication to occur, a primer need to be set first, okay? So a primer, especially when it comes to the Okazaki fragments, right? Or the DNA that's going backwards, okay? So a primer will tell the DNA polymerase, all right, you have to start here, okay? All right, so a primer would tell the sequence, okay, this is where you start, this is where you end. So you usually, in DNA polymerase, right, it doesn't tell you where you end. It does tell you where you start, okay? So it's going to tell you, okay, you start this way. This is what we call the forward primer, right? But the other side of the strand, okay, we also have what we call a reverse primer, right? When we go through PCR, that is when you guys will more or less understand why we do this, all right? So for now, I think I'll just, we're a bit behind on the topic, so the theory might be a bit like, right? But when we go through PCR next week, it makes a bit more sense because it's too long for me to talk about in this lab session. But you will need to create a forward primer and a reverse primer, right? In order to fully copy this template, okay? That specific template. So now, on this forward and reverse primer, you can design an additional sequence, okay? So of course, on the forward primer, it'll have to have ATG, right? ATG, okay? And this one has to have a stop codon, so maybe like TAA or something, right? There's 3 you could choose from, so all right, so it has to have that. But before those start and stop codon, you can code for the restriction enzyme site in the beginning, right, of your forward and your reverse primer. All right? So right now, that's what we're going to do. We are going to create the primers that will help us in PCR to take out this sequence. The tricky part about forward and reverse primer that gets people confused is that the forward primer is in opposite direction. So for example, your forward primer, which has the restriction enzyme site, as well as this, okay? This is going from 5' to 3', okay? Based on this direction. This one, all right, is going to be here, same 5' here, okay? But, okay? And this is going from 3' to 5', just like this, right? So this end is 5, this end is 3. So in other words, when you're writing this sequence, you have to kind of flip it around from 5' to 3'. So your restriction enzyme site, so if, let's say, I have my ATG here, okay, and let's say this is TAA, okay. So if you flip it around, it's going to be 5' to 3', It's gonna have your restriction enzyme site. It's gonna have your primer, right? But this is going to be flipped this way. So you have your restriction enzyme and your AT, okay? So it's kind of like mirror image, right? But the program will do it for you. But I'm just saying, right, we're flipping the primers in a different direction, all right? So now you have to create your primers. It's quite easy when you have this. So usually before I use benchling for the first year of my thesis, I would use like pencil and paper, okay? Because I think that time they haven't really had a really good program for it, or I don't know, I didn't find a good program. I usually just use my hands because sometimes I would like to insert certain sequences for better replication and transcription. So for me, I prefer using my hands, but this program makes it easier for people if you prefer to use it, right? Now, your OD sequence already have an ATG, lucky you, I added for you, right? And also a stop codon, right? And this sequence has double, so there's 5 to 3, and there is the 3 to 5, okay? Now, what I mean it's easy for you is because in order to make a primer, you would just click, usually primer, we don't want it to surpass 20 base pairs, right? When we talk about PCR, I'll let you guys know why. And you would right click this and you will press create primer. And there you go. You could click forward or reverse. You don't need to mirror image it or nothing like that. All right. You click forward or click reverse and it'll give you. But let's not create a primer. Let's not jump the gun here because we need to first choose our cutting site. All right. So I want you guys to go to each of these vector, okay, right now, right? And I want you to go, for example, PS, okay, let me see. PS, you have here the multiple cutting sites. I want you to look at the multiple cutting site, okay? We would insert it. So right now I'm just telling you where you insert your sequence in each of the plasmid, all right? So for PS, if you choose PS, you're gonna insert it into the multiple cutting sites available, right? And for PUC, right? PUC also has a multiple cutting site. You're gonna cut the LAX Z alpha. Wait, let me see if there's another. No, that's the only one. Okay. M13 river. Yeah, this is the site. M13 forward and reverse, guys, those are just universal primer we use to check. But anyways, so multiple cutting site is also available at PUC 19. That's where you insert your sequence, okay? So I'm telling you where you can insert your sequence, take a note. And then for PIGZ, you need your this promoter, right? You also have a sorry multiple cutting site available. If you look at this PIGZ alpha, you would know that it is for a expression of a protein because look, it already has a his tag for you. Lucky you, all right? So, meaning that you can do column affinity column chromatography, right? Okay, so here you have the MCS, all right? You can insert it. Just make sure you do not cut the promoter. So EcoR1 is the closest you can go, right? Don't use the BSCB1, right? That's a bit too close to the promoter, okay? And then for P-Cambia, this already, I was doing something here. For P-Cambia is a bit tricky, all right? For P-Cambia, let's see. Okay, for P-Cambia, there is a multiple cutting site, but I don't want you to use the multiple cutting site. Okay? I want you, where's my P-Cambia? Okay, I want you to insert, because I want overexpression, so I want you to insert behind the CAM V35S promoter. So you have to find a way, enzyme cutting site behind this. All right? It's a bit weird because it's circular, so it continues, right? If it ends here, it's going all the way up here. Okay? But when you insert, you have to make sure you're not breaking the Gus protein, all right, or any important promoter. All right? If you're cutting here, you're gonna disrupt the Gus protein. That's not gonna work anymore. Right? This also has a histone. Okay? All right. So I'm just telling you where you can insert. Now, the second thing to do, all right, the second thing you need to look at is if you can use any of these restriction enzymes. So go back to your OD, right? Look at this map. That's a lot of restriction enzyme. You have to make sure that the enzyme you choose do not cut your DNA sequence. If it does, then you will have a non-functional protein, all right? Or it's not going to be transcribed if you cut off the ATG, or it's going to have what we call a premature stop codon. Don't cut your DNA, right? What you can do is you can go to this scissors, all right? The scissors will tell you what are all of the restriction enzyme sites that are on, right? So see, you have here, this have 0, there's 71 of these, right? 7 of these, okay? So look through the OD sequence, okay? And see, right? What restriction enzyme site you have, okay? And you go to each of these vectors, right? And you choose, so the exercise is for you to look at the restriction enzyme site that is on the vector, right? And you have to make sure, okay, that the restriction sites on the vector is not on your OD sequence. That's gonna take you a while, all right? So I'm gonna give you a few minutes. You're gonna go choose, all right? For example, I can use FA as an example because that's not your sequence, okay? So for my FA, right, I'm gonna look at my sequences. I say, okay, these are the restriction enzymes that are here, right? Sorry, I selected these. Let me just deselect them, okay? So I'm gonna say, okay, you guys can choose any of these vectors, it's up to you. So I will say, unless those vectors do not have the restriction enzyme that your OD needs, then you move on to a next vector. So let me just choose PS2 first, since it's the first one here, right? And I'm gonna say, okay, I can choose any one of these, right? So I'm gonna say, okay, let's look at HIND3. All right, what if I use HIND3? So I'm gonna go to my FAA. I'm gonna go to my restriction sites, and I'm going to try to find if there's HIND3. So it's all in alphabetical order. Better for you. So you're gonna check HIND. It has a HIND3. There's one, so I can't use that. All right. So let's go again. ACC651. ACC651. Okay, let me check if I have that. So you check again. ACC651. Oh, I have one in my sequence, so I can't use it. Next, right? KPN1. So go to your sequence, KPN. Oh, where's K now? HIJK. KPN1. Oops, I have KPN1. All right, can't use that. ECO53K1. All right. So it is a time consuming. So I advise you to do it now because maybe you might not find in PS. You have to move on to PUC, pick Z, pick Cambia. All right. So find your restriction enzyme site. I give you 10 minutes. All right.
So you want we do it for each one of them, the PS and the rest of them one.
You just choose one. If you use PS and you could find one, then you could stay with PS. You can use PUC if you want. Start with PUC, right? Usually PS is when you want to express an yeast. Pig Z is for yeast. Picambia is for agrobacterium. PUC is for E. coli. But I'm not gonna ask you what organism you're in. I just want you guys to find the restriction enzyme site. I provided 4 just in case one of them doesn't work because I didn't check
Miss,
out the OB sequence.
where's the FA?
No. You guys are doing OD. I'm doing FA as an example because this is your assignment. So,
Okay, so we gotta compare it to the OD one.
mm-hmm. Okay, I give you guys 10 minutes. I need a little bathroom break. Drinking too much water. So find your restriction enzyme. We will 10, 4, 5, 57, 10:57 resume. If we're not in the lab bench, we're doing this. Okay. Be right back. I need my key.
Pcambia have for XBAI. It doesn't have, so I guess maybe that one could work.
Okay, are we done? Did you guys find your restriction enzyme site?
We could have picked anyone, right? Doesn't have to be in order or anything.
Yep, anyone, as long as it does not disrupt your promoter or your other proteins. Hi, what happened?
Okay, teacher class.
Okay.
Teacher Queenie.
Yes, I could. Okay, guys. Oh, are you guys finished? I did we at McDan find the restriction enzyme site? They're putting don. Okay, I see who done. Everybody done? Okay, I see lots of done, so I assume you guys are finished. All right. So the next step, if you find your restriction enzyme, right? I didn't even find my restriction enzyme yet, but I think, like I say, put not one. I know FA don't have that. LMN. Yep. Okay, I'll just use not one. All right, for my example, if I'm using PS, I'm going to select not one, NOT1, because, whoa, that's a lot of GC. Never mind, wait, I don't like this. Why I don't like this? Because that means my primer is gonna have a lot of GC. That's not necessarily a good thing. Let me check another one. I wonder if I have eco R5. Eco R5. Please not have eco R5. Damn it. Oh, it doesn't have eco R1. Yes. Okay. So I'm going to use eco R1 since it doesn't have that. Right? PS has an eco R1 here. Okay. So my restriction enzyme is going to be eco R1. All right. So the next thing you want to do is look at your restriction enzyme that you have, whichever one you use. My one is eco R1, which cuts the sequence GAATTC, and it cuts between G and A. So the restriction enzyme recognizes the sequence GAATTC. Wherever it has GAATTC, it's going to cut, right? You also want to check that it only has that one site there, because sometimes plasmid is supposed to only have one restriction site, right? Per plasmid, so you don't have multiple so much, but you have to be careful. For example, okay, all right. So I forgot to mention this important part to you guys. Part of the lesson. So look at your restriction enzyme right now, right, on PS, okay? So you only have that once, so you shouldn't have that site on your OD sequence. So now you observe it in your PS or PUC or PIGZ or PCambia, whichever one you choose, right? Now you're going to look at this sequence. How many cutting site is on
I
that
think
sequence? Yes?
some people are still
Oh, are still
stuck
doing it, finding it?
on it. Yeah.
Oh, okay. 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 All right, Dijonay. All right. So to look for the restriction enzyme site, right? You just need to click on this scissors, right? Which will, you could do the linear map, which is better to visualize, right? And then you click on the little scissors, right, to find the restriction enzyme. So the restriction enzyme should have a list here, right? Find the enzyme. Heck, you could even search it if you know the name, right? And what we want, or what I want you guys to do, is to find a restriction enzyme that is absent, the word absent, absent on your OD sequence, meaning it's not on your OD sequence. You do not want the restriction enzyme to cut your sequence as it is. Because if, let's say, I choose NSP1, right, as my restriction enzyme, it's going to cut my DNA in almost half, right, which makes OD non-functional, right, because it's not going to be a complete sequence. So you do not want to choose a sequence that is on your DNA template. What you want, however, is go to the different vectors. So I could click PUC, all right? And you could click on the little scissors on whichever one. So I'm giving you an option of 4. You could choose whichever plasmid, and you're going to click on the little scissors, right? And you're going to find a restriction enzyme that is present in your plasmid, but not on your sequence, okay? But here's the catch. But you guys need to understand that part first. So does that make sense to you guys? So for example, AAT2, right? Let's say look at AAT2, which would be here. No, why did I select that? Hold on. All right, so let's say I select AAT2, right? AAT2 would be highlighted here, right? So let's say I select wait, I don't want AAT2. I want something in this site. So let me see, where are these ones? No. Okay. I'll do it. So BSOB1, I don't think OD have that. No. So BSOB1, right, is an enzyme that I could use as a restriction enzyme for you guys' sequence. All right. Don't choose that. I don't choose it. For PUC, if you're doing PUC, right, because PUC have this enzyme restriction site. So it's going to cut around here, right, in the multiple cutting site area, okay? So that's the area where I ask you guys to insert your sequence, okay? So there I find one of the cutting site that you could use, right? Are we all there? Okay, good. Now, look at that cutting site that you have chosen. Here is the catch. Here's the catch. How many cutting sites are on the plasmid? Here, my one says 1. You have to make sure that it is only 1. If it's 3, if it's 4, if it's 5, right? So for example here, this one have 5. Okay, that's a lot. This is for another purpose. This is usually when we check the plasmid size. So this you cannot use for cloning. I would call it cloning. This you can't use for cloning. You got to make sure, right, it is only 1. You only have 1 of these sites. For example, this is only 1. It's available here, right? This as well, just 1. So it has to just be 1.
Wasn't it? I'm getting a bit confused now because I thought it was supposed to have none.
No. On the plasmid. On the plasmid. Because I'm in PUC right now. So on OD, it's supposed to have none. But on your plasmid that you choose, right, from the multiple cutting site, on your
So we're still not done selecting?
Yes, you're still not done selecting. So the next, like I said, the next step, you have to make sure there's only 1 of them existing on your plasmid. Of course, there's 2, 3, you can, but we won't get into that to avoid all that craziness. 1, okay, 1 cutting site. All right. You can choose 2 different ones, but I don't want to confuse you guys. All right. So just choose a cutting site that has one. I can make it more complicated. I can ask you to choose two that has two. But maybe in class we could discuss it. See, that's why in class is better. I could write it on the board. All right. This is only one method, okay? So don't think this is the only cloning method. No. There's so many problems you need in cloning. You need to think outside the box, right? But this is just for you guys to get used to the process. So just choose one cutting site, all right? For example, let me pick on somebody. Let me see. Where's the list of people? Myron, what is your cutting site and on which plasmid?
For PS and on the OD, for PS, I found the Eco 53K1, and on the
Equal
OD,
53K1. This one? Okay, that's only one that could work. And then on your OD, okay. All right, good. You could double check. How about, let me see, who else I could pick on? Jeremy, what is your restriction enzyme on which vector? Plasmid.
The PFO1 and from PYES2. PS2.
PS2 and which restriction enzyme did you say?
The PFO1 or that
PS
PF
oh TS?
PFOI, I think. PFOI.
I
That's
don't
the
find
name.
no PSOI though.
No, PS, PF. F.
Oh, okay, okay. PFOI. Sorry. Okay, good. All right. See what I? Okay, you guys. So far, so good. Pick phone. No. All right. Okay. So now that you have your restriction enzyme, right? Look at your restriction enzyme. I see my. What did I use? PUC, I think. Eco R1, I think. Okay. So look at your enzyme and look at the sequence, okay? I'm gonna go to my linear map. This is equal R1. My sequence is GAATT, right? It's DTG, sorry. GAATTC, and then the other one is, the other side is CTTA GC. Yes, I'm gonna copy that, okay? So look at your sequence. You could copy from, no, you can't copy from there. You have to copy it from your map. So copy the sequence from the equal R1. You're going to highlight the entire sequence of your restriction enzyme site. So my restriction enzyme site is GAATTC for equal R1. I'm going to copy GAATTC, right? You right click on the mouse, you press copy, something should pop out. You click on what it says DNA sequence. Just click on it, right? And you should be able to copy it. It'll say copy to clipboard, right? So see, I should be able to paste it like that because it's copied. Okay, so copy that, right? Now, easy thing. What I want you to do is go to your OD sequencing map, right? For me, I'm going to go to my FAA, okay? Sequencing map. You got it? Go to your sequence map, copy So first, copy your restriction enzyme sequence. Now go to your sequence map. I want you to right click.
Miss,
Well, yes,
the cut starts where the black line is, or the highlighted area.
the highlighted area, right? So if, for example, here, this highlight, even though the cut starts from G or C at the bottom strand, right? It starts at G, GC, GC, okay? It has to be the highlighted area because that's where the restriction enzyme recognize. So it will recognize those four, for my one, 4 base pair sequence for HHA1, right? So you have to copy the whole thing that's been highlighted, all right? Everything. Let me know when you guys are good to go. Good? Copied? Make sure it's everything, you know. Ah,
Excuse me, miss, can you
yeah.
please repeat where we ought to input, or are we just doing the copy so far?
Copy so far. All
Okay.
right. So,
Sorry.
so for example, right, this one, you'll have to copy all 4 of those base pairs, GC, GC. Everybody done copy?
We're doing your one or our one? Our one.
No, no one. You're doing OD.
Okay.
You don't have FAA, so you're doing your one. Where whichever restriction enzyme you choose, that's the way you to do. I'll check them later. All right. So now you want to insert those sequences in front of your ATG. Okay. So you're going to right click, put insert bases. Let me. Does it show like the other window too if I click it? Insert bases, right? And you just paste. Right, so this is my EQR1. I want to insert the 6 bases at position 1. So you press enter. There. Right?
Where do we go? The OD.
Yeah, you guys have to go to OD, right click on before ATG and press insert bases. And you would insert the sequence for your restriction enzyme
So
site.
it doesn't matter where, it's gonna automatically.
You have to put it before ATG. So you just paste it, right? And you enter it. And then it should, for example, here, see, it should be able to give you that sequence.
Miss, so just post it right in front of the ATG sequence, right?
Yes. Right in front of the ATG sequence. Got it? Because you still have to paste it somewhere else. Oh my God, who is calling me now? Hello? Cabbage sandwich? Oh, I want to eat the chicken salad then. Yeah. With what? Cranberry green tea. No sugar. Yes, please. Thank you. Okay. There goes my lunch. All right. Okay. We ready? I don't hungry. All right, guys. So after inserting that, that is your forward. Okay. You have to do the reverse. So you have to go behind your, for you guys, OD sequence, right? After TGA, you're going to insert this same cutting site, all right? Restriction enzyme site. So do the same thing, okay? Insert bases, enter, all right? It should give you, wait a minute, TAA. Yeah, that's fine. TAA. Let me check. TAAGAAT. Just gonna be before ATG. Yeah, okay, that's fine. All right, so you're gonna put behind, okay? So before and behind it, okay? Before and behind, all right? GAATTCA. G. What does it call? GAATT. A A T T C. So it's gonna be G A T T C.
Is there a way we could remove or undo
Ungroup?
undo?
Yeah, you could undo CTRL-Z. You could press CTRL-Z and it should undo. Well, that's how I undo. Yeah, that's CTRL is control, yeah. Control-Z. Finish? Let me know when you guys are done. G, C, T, T, A. I'm just trying to draw my diagram. A.
Yes. So for clarify, we supposed to put in the reverse or the say
The thing
sequence.
is, this program is going to do the reverse for you. So all you have to do is paste, right? Like I said, it's not
Yeah,
gonna, yeah. So you just have to paste the same thing. It's going to recognize what site it is and put the reverse for you.
that'd be at the end, right?
Yeah, at the end.
Okay.
That's a good thing with benchling. Now I have to do like them old time method. I didn't know they had that, so I had to freehand it. A T T C G. This will be G C T T A A. Oh, oops. Let me know when you guys are done so I could share this. A T G T A C T A
So we're just
A.
gonna add them, and that's it for right now.
Yep. Okay, done? All right, guys. So basically, what you're doing or what we're doing is so this oleate desaturase sequence, okay, this DNA sequence is found in plants. So what you're doing is we have to make a primer to remove that specific sequence, right? I think your OD is around, how much is it? Is 1,152. So you got to remove 1,152 base pairs from the plant's chromosome. And in order to fish that out, we need to do PCR. And so PCR, the first thing to do is to design these primers, right? So we need to design the primers so the primers will stick onto the DNA sequence on the plant's chromosome, and the polymerase will be recruited to that specific sequence and copy it. That's why you need a forward and reverse strand to tell the polymerase exactly where you want the sequence to be copied and copy that over and over through PCR. But why we add a restriction enzyme is that other than the sequence, right, other than the ATG blah, 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 so for your OD, so other than this beginning sequence, okay, so usually the primer would be the beginning and the ending sequences. So other than the beginning and the ending, we want to add this restriction enzyme site so that when PCR is happening, it not only copies the ATG and forward or the TGA and reverse, it also copies the restriction enzyme site that is on our primer. And so what happened is the end product of PCR will be this entire sequence plus the overhang, which are our restriction enzyme sites, all right, or restriction enzyme sequence, okay? So in order to make your primer now or have a sequence for your primer, you're gonna select your the beginning of your sequence. So your one will have the restriction enzyme site, right? On top of that, you want to select about 20 base pairs. The reason why we don't select many base pairs is because RNA is known to form secondary structures, right? So these ATGC and all of these things, they might look linear right now, but we know that they're capable of hydrogen bonding. The longer the strand, the more they can stick onto each other. If they stick onto each other, PCR might not be as efficient. So you don't want to have that long strand of primer, right? But the primer cannot be too small as well, because the smaller it is, the less specific it is. So when you do a PCR with a very short primer of maybe like 5 or 10 base pairs, it's gonna stick onto random other sequences on the plant genome, right? Because the likelihood of having those small little sequences is higher than having 20 base pairs that are the same. So that's why 20 is kind of like the lucky number. Sometimes we can't always have 20. because of other parameters, which I will show you right now. Okay, so you select the first 20. Yes. All right, no problem. So you select the first 20 nucleotide sequences right now, right? So I'm gonna use your old OD without restriction enzyme site as an example. So I'm gonna select 20. At the bottom, you can see there's some numbers. So it starts at 1, end at 24. Basically the length, right? So just how many you selected. So I select 24. So I'm gonna select just when it reaches 20, there you go. So I'm gonna select 20. I will add and minus based on the parameters, right? Yes, for you guys, you include your restriction enzyme site. I didn't stick it for OD, right? So please include a restriction enzyme. That's the reason why we did it, right? So your primer can have it.
Where do you see the count?
Right at the bottom here, where my mouse is. So here it has start, end. The length is the count, right? So you have like 20 base pairs. Now, oh, you don't even have to click it. Now, beside the length, you have something called the GC, and you have something called the melting temperature. Remember, we talk about TM or melting temperature in class, right? The melting temperature is the temperature where the primer, because primer also comes double-stranded, you know. Hold on, let me think about it. No, actually, they don't come double-stranded. Sorry, they come single-stranded. But the melting temperature, what happened is that these strands, they can, like I said, kind of stick to one another based on the GC content. The more GC you have, remember, the higher the melting temperature. So you have to have a kind of equilibrium, if that's a word to describe it. The GC content will be best if it's below 50%, but cannot be too low. I think 50% is the ideal one, around 40 to 50. So 65 is a little bit too much. I have to see how I can adjust it. Sheesh, that GC is high. Are you sure?
Excuse, what the GC do again?
The GC content is the percentage of guanine and cytosine on the template. So the ATGC, how much G's and C's you have, right? And we talk about in class that GC triple bond to each other. So what that does is it's harder to break those triple bond. And so if you have a lot of GC content, it's harder for you to kind of break the secondary structure that these single-stranded DNA will have. So what you want to make sure is your GC content is not too high, but also not too low. All right. So a good GC content will be around like 50% GC content. Melting temperature is good around, I think 58 is fine, 55 is fine, but you don't want it to reach 70 because that is where your polymerase start copying. But definitely melting temperature has to be above 50, about 45, 50, right? So you have to, if, for example, you can't get it below, well, that's just how it is. You can't help it, right? So see if you could find the good GC content. Although, you know, I don't like, I don't really use the one here. It's not that accurate. I use another program. But this is looking at both forward and reverse. So probably that's going to give you some false. So what you can do is you can click 20, right? Even though it's giving me 65% and 63.8 degrees Celsius, I can check with just looking at the forward because that's what we need. So I'm going to right click after I choose 20, right? I'm going to right click. I'm going to press create primer and I'm going to click on forward. So click on forward and it should give you a window like this. All right. So, um, when I give you this window, it will give you your TM, right, and your GC content. My TM is too high. Wonder if what will happen if I remove it. Oh,
What would be too high?
You don't want the TM to go above 60 degrees Celsius, but like I said, because we are fishing out primer, if you cannot get it below 60 degrees Celsius, it's fine. If that's the lowest you could go.
You're at 56, right?
56, you can decrease the number of base pairs, but again, if you are at 15 base pair, that's where you want to cut it. You don't want to go any more below. You might have random PCR product because your primer is sticking to all kind of sequences. So you want to make sure your sequence is above 15 or at 15, and you want to make sure annealing temperature is not above 60. You want to make sure that the GC content is around 50 is the best. You could go a little higher, is fine, but too high is not good. Right? So GC content 50 is
So you
perfect.
could have a good sequence, cutting sequence, but still not. This still doesn't work out properly. You're not gonna get what you need, or
Yes.
you have to start
So
all over.
okay. Good question. So when you're creating your primer, right, we try our best to match the perfect parameter so that it sticks onto the specific sequence like almost 100% of the time, right? And it does not stick to other DNA we do not want it to stick to. But when you're fishing out a specific sequence, you cannot change your primer too much, right? So you still try your best. If it capture other sequences, and we'll discuss this after PCR, is what you will do is on your DNA. So if I could draw a DNA gel. On your DNA, you have your ladders, right? And what's going to happen is you might have the product that you want, but you might also have other sequences along. And it makes it a bit difficult for you to know, all right, or for you to get a pure product. If in that case, it's fine. What we usually do is we look at the number of base pairs we have. For example, the ladder usually goes smaller. So maybe you have like a 1,000 here. You have like 500 base pairs here, right? If let's say my sequence is around 500 base pair, I would know that this is most likely my sequence. So what I would do is I would cut out the gel and I would isolate this particular sequence. I will purify, right, the DNA from this gel. I will send it to sequencing just to double confirm, right? But we don't like it when we have all of this other byproduct of PCR. So one of the things to ensure that doesn't happen is to make sure that the primer have almost perfect parameters, right? So that's why we check the annealing, we check the GC content, and we also check the length, okay?
So you don't have to start over. It just makes it a lot more, I don't know, clear, or precise.
Yes. It makes it more precise and you don't have to do, a whole other cleanup work, we call it. So if your primer is good, right, you would only see one, on your DNA gel, you'll see one band, and you could use that to continue your work. You don't have to purify it from the gel. You don't have to dig it. You don't even have to go on. You can go on double confirm with sequencing, but usually we would know that is the product, right? And you only have one band. So it just makes your life easier when everything goes perfect. But of course it doesn't, right? So that's how it goes, guys. That's how it goes. All right. So you guys make your primers. So my GC content is still high, but I have an okay annealing temperature. I have around 15 base pair right now. I left it as it is, right? I will name this forward primer. Okay, so I'm gonna, oops, already exists. Which one is this? OD. So I'm gonna name it forward OD, right? I'm gonna save it, and it should appear there. Sorry, my I don't know what's happening with my bench. I think
So
it's
it's kind of added before now, the primer. Well, not sorry, not before. Yeah, before.
is added before.
Yeah, yeah, because you're adding this before the actual what would you say? The forgot. Is it the cutting
PCR.
sequence? Yeah.
Oh, before you do PCR, you need to know what are your primers, right? Because PCR require primers. So before you do any kind of PCR, you need to first plan. This is what we usually do before we do any experiment. We need to plan our experiment, right? So first you need to design your primer. And in order for you to design your primer, you need to, number one, know what is the sequence you want from the organism. In this case, plant. The sequence we want is OD, right? And in order for us to get that primer, we need to know, okay, we need a forward and reverse primer. Our forward is at the beginning of the sequence, our reverse is at the end of the sequence. So you have to design the primers. And what happened is, after you design the primers, we will send this particular sequence you design, you send it to a company which basically builds the base pairs for you. All right, so you could design, this is how you would design your primer for PCR, right? Yeah. So for you guys, make sure you have your restriction enzyme site, okay?
Miss, so what you do for the forward, you do for the reverse too?
Yep, what you do for forward, you do for the reverse. So my benching going weird. This is supposed to be up here. Just forget about that. I don't know what to go on there. But you would
20
choose
also.
your, sorry?
20 also. Same
Around 20,
parameters.
yes. So remember to click reverse though, right? So the program can reverse it for you. Right? 57 is fine. 33. Let me see if I could save it. But that's a
We
lot of
gotta
base
we
pair.
gotta subtract it to get the 20 though.
We no. There goes my. Okay, that's fine. 45 is good. 35 is near 50. I will take it. Okay. That's gonna be my reverse primer. Okay. I don't know what's happening to my probably because I have two of them open on one computer. No, watch my bench thing. This thing supposed to be all the way here, but I don't know why it's pushing it there.
Miss, I
So,
got a question.
mm-hmm.
What does the check secondary structure at like the certain degree Celsius do? Like, what does that feature do in the program?
The check secondary structure.
Yes,
The
just
checks. Okay, sorry. The check secondary structure, like I said, each of these primers, right? They're single-stranded DNA. And usually DNA is double-stranded, right? They like to form hydrogen bond. So what you can do, if they can check secondary structure, they can, I don't know if this program, I have another program for primer making, which will actually show you the different types of secondary structure that can be formed. So I'm not sure if this program also shows you the different secondary structure. So they form what we call a hairpin structure or pin loop structure, right? So if you have a single strand like this of DNA, what can happen is certain areas can stick together like this. Okay, this is what we call the hairpin structure, or they could be like this. Right? So certain areas, they could stick together based on the base pairs that's there. Because we know A binds with T, G binds with C, right? So if they find each other, they might just hydrogen bond to each other, forming these secondary structures, okay? So you could check it. You could run the program and see what type of secondary structure shows up. Or they might give you a value. But I personally I don't really use their primer making and primer checking. I have um, my own way of doing it. So and then you can check. Yeah. Okay. Yeah. See, they give you a value. Like I said, homodimer, meaning that the sequence can stick to each other. Okay. Or a monodimer, meaning that they can have these hairpin loop structures. I have another program that actually shows you all of the homodimer and monodimer. But that's for another story, somewhere else. Because I didn't really use benchling much, but it's a good program. I mean, it's a good website. So this can tell you, okay. Oh, there, they show you actually. Oh, wait, I'm not even sharing this tab, sorry. So, so they can show you, right, what is the homodimer. This is when the two forward primer would associate with each other, right? Or you can have monomers, but there's no monomer. So they don't want to stick to each other, the single strand, but the two strand, they can form a homodimer. Okay, so this just shows you that the likelihood of them forming this, but that's why you heat them up to their annealing temperature so that they can, not annealing temperature, melting temperature, so they could split, right? Okay, we're at 11:39. Sheesh, just to make primers. Okay, so, woo, okay. So after making your primer, all right, after making your primer, now what I want you to do is go to your plasmid that you have chosen, okay? This part is the tricky part. Go to the plasmid where you have chosen. There will be a site, for example, if I choose eco R1, all right? What I want you to do is delete those sequences and insert your sequence, okay? So for example, let me, why not the Makai do it? What? No, hold on. Okay, moving on to another folder. All right, so what I would do is I select eco R1, I think, yes. Okay, so what I would do, or what I want you guys to do, select your restriction enzyme site, right? You delete the restriction enzyme site. So for example, eco R1 is GAATTC. I'm going to take away GAATTC, okay? Delete. So to delete 6 base pair at position 396. So I gotta make a note, all right, that the sequence was between CAGT and GAGC. All right, just making a mental note. All right, it's between CAGT
Miss, can you
and
zoom in a bit closer to,
oh,
because
okay.
I'm trying to see
Zooming.
where you're identifying where it was cut.
Okay. All right. So this is my Eco R1 site, right? It is GAATTC. So GAATTC. GAATTC is in between CAGT and GAGC, right? So I'm going to delete this site, okay? Make a mental note of where it's between because that's where we will insert our sequence. And we're going to press enter. Bam, equal R1 disappear. All right. Then you're going to take your OD sequence with your restriction enzyme site. I'm going to insert my FAA since that's what I have. All right. Oops. All right. Copy the DNA, go to this, insert bases, right? Paste all of these bases, enter. There you go. So now you enter the sequence, right? And what you will find is that you will have eco R1. Well, for me, eco R1 is at the beginning, right? And at the ending, okay? Which makes sense because if you look at our explanation before, right? If we cut open our plasmid like this, if you cut open your plasmid, right, and you insert your sequence that will also be cut, it will create the same sequence on our vector, right? On our plasmid, meaning the beginning sequence will have your restriction enzyme site before your ATG, and the stop codon. After the stop codon, you will also have your restriction enzyme site, right? Which is the same here. So in the end, you have your stop codon TAA, and then your restriction enzyme site. In the beginning, you have your ATG here, ATG, and in front of it, you have your restriction enzyme site. So what happened is when you do what we call an enzyme digest, right, and you cut this plasmid and this enzyme using EcoR1 restriction enzyme, it's going to cut the sequences in this way, right? It's going to cut the sequences here, all right? So this is for your plasmid. It's going to cut, okay? And on your PCR sequence, it's also going to cut the restriction enzyme site, which is this site and also this site. So when you anneal the DNA, right, you're going to anneal the template onto the plasmid. So basically, you cut out the plasmid and you insert the template, okay, using your newly created restriction enzyme sites. All right. So
So you
you should
select
have,
the option that says cut base, delete bases.
yeah. So you would select, for example, right, you will select EcoR1 site on your, or whichever restriction enzyme site, and you just press backspace, just delete, right? Backspace, and it should ask you to delete, and you delete that. And in its place, you will enter your sequence. Okay, so try that. And then we have one final thing.
Miss, when you copy the sequence of the OD, you have to copy everything
Yes,
from.
everything from restriction enzyme to restriction enzyme site. You could do CTRL-A, right, which should be everything, and you just copy it, right? So everything that you have created thus far, right? If you realize that 22 is better, 2 base pairs is fine. All right, so you could do the 2 base pairs. What I usually would do is I would check the secondary structure. But I don't think 22. I've made a primer with 30 base pairs before. Got scolded by my PI, but you can do the 22. It's fine because it costs money. Every base pair, I forgot how much money is every base pair, but they count by base pairs, right? So the longer it is the expensive, the more expensive it is. So you have to also account for that. We done? Okay, I'm waiting. Don't worry. We have like 15 more minutes. Then I could go for lunch. And I will I will show you how you can share with me so I could see what you've done. I wonder if I had a chance to know with the assignment. Like I see what the assignment says. Oh, oh, oh. Insert your gene into the plasmid vector map. Yes, you guys did that. Okay, sorry. Miss, what time will you be in office so I can drop off our group's lab after lunch though? After lunch, I'm here until 4:30. I think I'll be here. Yeah, so between like 2 to 4:30. If I'm not in this office, they might call me to go in the chair's office, so I might be with Miss Karen. So if I'm not here, I'm over there. Um, I'm always here, guys. I'm always here, unfortunately. Unfortunately, I don't know. All right, we're finished? Oh, gosh. Okay, well, just make sure you remember this part then. Okay, so after that, I want you to visualize your sequence, okay? So my sequence, so you want to visualize it and maybe want to categorize it. So you're gonna copy your sequence after your restriction enzyme. It starts from ATG, right? So it should go to, where is my end of my other sequence? Oops, we don't lost it. Where's my equal R1? Somewhere here. Here. Okay, TAA. All right. So I like to visualize it. I can say create, where is it? Create amino acid sequence. So I want to create a amino acid sequence, okay? You could also create an annotation, right? So I can create an annotation. I can give it the name for you guys is OD, FAA, right? It is a coding region. Okay. I give it to a pink color, I guess. It's a forward strand. Okay. So I can annotate it. Okay. So there you go. So when you go to the plasmid map, you'll be able to know, okay, I have inserted this sequence in this direction, right? So the promoter is going to be, well, I have no idea. There's no promoter here. The promoter was this way, but I went this way. Okay, so I've inserted this sequence, okay? And you can also create an amino acid sequence for it. No. No, I don't want translation. All right. Okay. Oops. Okay. So I mean, you can look at the amino acid. They'll give you some properties. They will even give you percentage of this, your net charge at different pH. Really good. You can predict the 3D structure as well. I mean, nowadays they're
Yes, can you
updating.
repeat that one more time, please? Like
Oh,
the part where you copy and you pasted it and then you went a little too fast.
I copied and pasted it. Oh, you mean copy the entire sequence?
Yeah.
Okay, we could copy the sequence. All right. You know what? I will destroy my PS. Okay, let me use PS. I hope eco R1 only have one side. Okay, we'll use this one. All right, then fine. We will do one more example. Okay. So for example, if you want to insert your sequence, I'm going to use eco R1 again because that's my FAA sequence, right? And it only has one restriction enzyme. So it's GAATTC. I usually write it down just in case I forget. Surrounded by GTC and also TGC. Sorry. Okay, so I'm going to delete this sequence, eco R1, GAATTC, enter. Hello. Ah, there you go. Wow. Wait, gone. This is freezing. Oops. It gone weird. Okay, let me do it again. GAATTC. Okay. By 500, around 550. So delete that. Enter to delete. All right, there you go. Now, in that, between those two, around 5,850. I mean, sorry, 550. I want to insert my sequence. So I'm going to take my FAA, CTRL, Control Alt, A, copy. Okay. Or you could right click copy, DNA sequence, go to my PS. In that point, right click, insert bases, paste my base. All right. And then press enter. Bam. All right. Here's your eco R1. And then you have ATG. At the end, here's another eco R1. You have your TAA, sorry, which is your UAA, which is your.
Can you explain how you visualize it? You said you could
visualize. Oh, with the amino acids and the annotating. Yeah. Okay. So for me to annotate it, you don't want to include the restriction enzyme site, right? So you want to start at ATG, which is the methionine that starts translation. And then you want to go all the way to the end. So that'll be around my eco R1 at the stop codon. Okay, TAA. So there's two things I want you guys to do. First, right click. There's something called create annotation. Okay, so you click on create annotation, right? You can name this. So we're trying to annotate this particular sequence. I will name it FAA since that's my one. You guys are OD, right? The type, it is a CDS, right? It's a sequence. Sorry. You pick a color. I pick pink because it's cute. It is a forward strand. I'm going to leave as forward because it's going to copy in this direction. Okay. And then you just press save, right? And it should show up here. Okay. So see, It's going to be from here, ATG, all the way to the stop codon. If you go to your plasmid map, you will see that it is here, right? My FA is here. It's before my GAL1 promoter, and it is before my terminator. All right, so it's a
Can you
very good
show where you added? Because um,
where I
where you selected from, sorry.
where I selected my
Yeah, where you began your selection and ended it.
oh, I begin to select from ATG. So from
What?
your restriction enzyme site, right after should be your ATG, right? Your start codon. So you begin selecting from your start codon because that's where the protein begin translation, right? So you will begin from the start codon and you will end at the stop codon, which is here, TAA, which is also before your restriction enzyme, okay?
Oh, you have 2 minutes.
So after that, you guys got it? That's one part of your assignment soon finished. See, easy. The next part, we'll have to figure it out. Okay, now, yes, there's a next part. So you can also create an amino acid, right? Sequence. You can just go to create amino acid sequence, press the forward, name it. Just gonna name it FA protein. You don't need to choose a scheme. You create, right? When you create it, it naturally gives you a lot of different options that you could look at, okay? Right? But it should look like this. So here, correct, ATG is supposed to be your methionine. That starts translation, right? And then in the end, if you have a little asterisk, that is true because that is the stop codon. Stop codon, they will usually put a little asterisk, meaning that that's it. Done. Ribosome is leaving this site. Later, hasta la vista, dissociate, okay? So now you've created what your vector is supposed to look like. Okay, so this is what your vector is supposed to look like with your DNA that's inserted, if you've done it correctly. Okay, so that's one part of your assignment. Now, I hope everybody got that.
Could you
Okay,
show that last part?
what happened?
The last part, because yeah,
The amino acid part?
from when you switch to this window.
Oh, because I just switched to the sequence map. So this is the sequence and plasmid. So once you
Well, how did you get all the amino acids?
on the sequence,
Yeah,
you
because this procedure showed us only gave us, for the insert.
mm-hmm. So what you would
Is that
do
a
is,
part of the assignment, or if not, then
well, you have to learn. I will give you points for it. So but you just need to right click, right? And you just need to create amino acid sequence, and that's it. So you just click
okay, thanks.